Assalamu alaikum my dear students. Hope you are fine by the grace of Almighty Allah. Welcome to NIEVS Home School. I am Nasima Akter, Assistant Teacher of Science, National Ideal English Version School, Morning Shift. Dear students, today I am going to take a biology class for class 10 students. Dear students, our today's topic is Chapter 12, Part 2. Let's see our today's topic. Students, our today's topic is Chapter 12. Part 2, and this is lecture number 7, and our today's HW number is 7 number also. So dear students, let's begin the class. Students, in our previous class we have learned about DNA. Today we will learn about RNA, and this is from page number 256. Here is written RNA stands for ribonucleic acid. You can underline this line, ribonucleic acid, RNA. Most RNA contains a single polypeptide strain. That means they have single strain. It is composed of 5 carbon ribose sugar, inorganic phosphate and nitrogen based. And here is the nitrogen base here is adenine, guanine, cytosine and uracil. Instead of thymine, here is uracil. In the DNA we have seen here was adenine, thymine, cytosine and guanine, but in RNA we can see here is uracil. In the gene of RNA virus, RNA is found as a permanent component or the inherited component. In some viruses, tobacco mosaic virus, DNA is absent. The viruses which are not composed of DNA possess RNA as their nucleic acid. In this situation, the RNA serves itself as the heredity material. Now, gene. The unit of controlling all the visible and invisible signs and characters of organisms is called the gene. Genes are positioned throughout the chromosome of organism. The location of a gene throughout a chromosome is called the locus. For specific characteristics, a gene is responsible. In some cases, a characteristics is expressed through cooperative action of several genes. In other cases, one gene controls more than one characteristics. From different research, it is clear that a gene is the perfect master of heredity. By taking necessary signals from DNA, RNA enters into the ribosome of the cytoplasm, and according to that signal, protein is synthesized. And in the case of a eukaryotic cell, this protein is first deposited in the endoplasmic reticulum, and then it passes through the Golgi body and vesicle. Meanwhile, the protein undergoes many changes and it carries to necessary places. In the prokaryotic cell, protein directly reaches the destination. This protein determines the nature and speed of biochemical reaction and form that the characteristics of living organisms are determined with respect to the environment. These characteristics include everything from structure to the behavior of any living organism. It can be expressed as DNA to RNA, protein and characteristics. The number of genes in different organisms is not the same. In organisms of the same nature, the number of genes is more or less same. Genes are arranged in common principle alongside from one end to another end. Chromosomes are separately spread from each other in a linear form. Different replicas addition of the same genes can express the same characteristics in different levels. For example, in a pig, for tallness, the height determination gene is T, and for shortest and the height determination gene is T, small t. Then capital T, small t is combined, it express its tallness. So capital T with respect to small t is called the dominant gene, and the small t with respect to the capital T is called the recessive gene. When in an organism two genes are recessive, only then does the recessive gene express the recessive characteristics. Only P plant with small t, here is 2 small t, is short in height. If we express here, then look at here, here is a two capital T and capital T, that means parents chromosome here is both capital, and capital T here is dominant character. From here, here is the gamete, two gamete, they are parent. If we cross them, then we will get here two types gamete, and here is small t, they are recessive. If we cross them, then we will find in F1 generation, here is one is capital T and another is small t, and again if it will cross with this, again it will be capital T and small t, it will be tall characteristics. And here is capital T and small t, and again if it will match with this one, capital T and small t, that means the whole F1 generation will be tall, because here is the T, capital T is dominance character and the recessive is T. When recessive, when we will get only these types of chromosome, then it will be short characters. Now if we cross this capital T or small t parents, then from here we will get, they are parents, we will get two types of gamete, capital T, small t, here is the same way, two types of gamete. If we cross them, then we will find here both are capital T, that means tall characteristics will be expressed, and here will be capital T, small t. For the presence of this capital T, it will be tall characteristics, and if it one will cross with this one, again it will be tall characteristics. And then when here is small t, both are small t, that means they will be short characters. So we can see, if we see this line again, when in an organism two genes are recessive, and we have seen that they are small t, only then does the recessive gene express the recessive characteristics. Only P plant with small t is short in height, that means here is 3 plant will be tall characters and it will be only 1 will be short, that means 3:1, and this is the first Mendel laws, and it follows the Mendel first law. Now to see how the characteristics of traits of parents are expressed in the next generation offspring, scientist Gregor Johann Mendel did some experiment with pea plants in 1866 and discovered a valuable theory of genetics. The fact that the presently glamorous from known as the gene. Gregor Johann Mendel is called the father of gene. Mendel took a tall and a short pea plant and transferred the pollen grain of a tall plant into the stigma of a short plant. In the same way, he transferred the pollen grain of a short plant into the stigma of a tall plant. This resulted in reproduction. He made sure that the other pollen grain would not appear. The reproduced seed were showed, and it was found that all the plants were tall in size and no plants were short. We have already seen in F1 generation they were all tall. It was found that there were both tall and short plants. Among them 3/4 were tall and 1/4 was short. This theory of Mendel is applied in breeding of plants and animals. The controlled sexual reproduction is done in plant and animal to bring about desired characteristics. As a result, a generation with various characteristics are produced. From this generation, desired characteristics are selected for breeding to get plants and animals that have desired characteristics. At present, this theory is applied largely for the production of improved variety of crops. Now, here is the DNA replication. Through the process, a new DNA molecule is synthesized from an existing molecule of DNA. DNA replicates in a half conservative way. Through this process, the double strand DNA becomes single strand by breaking hydrogen bond. Now come to here, this is page number 259. Then the floating nucleotide in a cell creates new complementary strand by combining A adenine with the thymine, T with A adenine and C with guanine, and the guanine with C. Thus one of the old strand remain and combine with a new strand to make a complete DNA. At the end, a new strand combining with another old strand from the structure of a molecule of DNA. As the new DNA emerged, it has a new and an old strand. You can see here is the DNA replication. In this here will be one strand will be new and another will be old strand. This principle is known as the semi-conservative method. In 1956, Watson and Crick first successfully proposed the replication process of DNA. Now, DNA test. Throughout the present century, DNA technology and the significance of it in the field of medical science, agriculture, fish and animal resources, and in the industry of medicine have introduced a new chapter on creation. In Bangladesh, along with the evidence witness, eyewitness depend in judiciary system, DNA has opened a new window of ensuring true justice. The scientific and practical method of DNA test is called DNA fingerprinting. The name DNA typing or DNA testing is also used to perform the DNA test. The first requirement is an organic specimen, bone, teeth, hair, blood, saliva, semen of a person can serve as valuable organic specimens. The profile from the place of occurrence of crime or the profile from the specimen of the victim of a crime is compared with the profile obtained from the blood or organic specimen of a. In this method, DNA is isolated from the sample by a chemical process, and then DNA are cut into pieces by one or more restricted enzyme. Then through a special method, the species of DNA are sorted according to the length of their different bands. Then radioactive isotope DNA is hybridized, keeping it on a special cellulose paper and then putting it on an X-ray film to determine different visible bands by autoradiograph method. After that, the sample is compared with DNA map of the victim or suspected person. This method is termed as a fingerprint method. Currently, by using polymerase chain reaction PCR method, DNA test is done with even a small sample for identification without any error. Now next is here determination of human sex. In human and other mammal, sex is determined in the same way. In the cells of the human body, the number of chromosomes is 46. 23 pairs, among them 22 pairs, total number here is 44 are autosome, and the remaining pair is the sex chromosome. Autosome plays role in the physiological and the embryonic development and the formation of the body of organisms which have no part in determining sex. The two sex chromosomes are marked with X and Y. They play a significant role in determining sex. If female, in the diploid cell, both sex chromosomes are X, that means here is 2X. In male, the two chromosomes, one is X and the other is Y. Both the chromosomes are long in structure and rod shaped, but Y chromosome is shorter than the Y chromosome. And look at here, the Y is shorter than the X chromosome. At the time of the development of the egg, meiosis occurs, and every egg possesses an X chromosome along with other chromosome. But in case of male, at the time of the formation of sperm, half the number of sperm contain an X chromosome and the other half number of sperm contain a Y chromosome. An egg can be fertilized with either type X or Y. Only one sperm X or Y of father successfully fertilized with the mother X bearing egg during gestation. So the sex of the future child depend on which sperm fertilized with egg. So a zygote can have both chromosome as type X or can have one X and the other Y. The baby which is born having both chromosome X, that means here is XS, will be a baby girl, and the baby who is born with the chromosome of type X and the other one will be Y, will be a baby boy. The determining sex, a mother plays no role because the mother always produced eggs containing only an X chromosome. On the other hand, the father produced sperm having both the type of X and Y. And look at here, the father chromosome here is XY and the mother chromosome contain only same types chromosome X and X here. If this X chromosome will be fused with this X chromosome, then the baby will be girl, and if Y chromosome will be fused with this X chromosome, and the baby will be boy, that means XY. Students, now I will give you HW. I have finished today's topic. Now the HW number is 7 number HW, and your HW will be make 5 knowledge based question. Your HW7 will be make 5 knowledge based question. So dear students, I hope you have understood today's topic, and keep practice at your home and do your assigned HW in your HW copy.